Hey everyone, let's finish this class with chapter 3. The most important part, I will say, adaptive immune system. Pretty important to know how why this system is so important. And it's very important also to recognize the two different functions of these two type of lymphocytes, B and lymphocytes T. And then after that, we're going to cover what happens when your immune system overreact, when your immune system is reacting against something that it shouldn't, right? Or that at least it shouldn't react that with such a extreme response. That's what we call hypersensitivity. This is very high yield to know. And also some autoimmune disorders, which now at this point you know that autoimmune means that your immune system is attacking your own body tissues, your own organs. And finally, we're going to finish with a little bit of information about cancer and organ transplantation. All right, this is the part that I tell you that many students is a headache for the students. Even I remember me when I was a student, I have a hard time with this part. But in reality, it's all about your mindset. If you have the general idea, you're going to learn it. It's not hard. I'll do my best to simplify this adaptive immune system. First of all, make sure you always think about innate and adaptive immune system, and keep in mind that every time you encounter a pathogen that enters your body for the first time, your body will have no idea how to fight that infection. So you will activate your innate immune system, which will lead you to have symptoms of disease. So you will have an infection. But the ultimate goal of this is to activate your adaptive immune system, because you're going to do two things when you successfully activate and develop an adaptive immune system. And this is what you need to keep in mind. First is activation of the adaptive immune system is the part of the immune system that will get rid of the infection. And more importantly, adaptive immune system will generate immunological memory. What that means is that if you encounter the same pathogen again, you will know exactly how to fight that microorganism and how to neutralize it pretty quickly. So the whole goal of the immune system is activate the adaptive immune system, right? Because it's very specific and is very efficient to killing or destroying whatever caused the disease. So first, we divide according to the time of exposure, adaptive immune system, and primary and secondary. Okay, so primary, the way how you activate this primary adaptive response is with your innate immune system. Okay, so unfortunately, when you have a primary adaptive response, you're going to develop symptoms. And those symptoms are always relatively severe. You're going to feel sick, you're going to feel terrible because the inflammatory response, because the fever that causes malaise, body aches, if it's a local infection, maybe signs of local infection like the ones that we review, redness, heat, pain, swelling, those kind of stuff. So, and I'm going to explain this in just a minute in the next slide. So always we have this primary adaptive response when we encounter a pathogen for the first time. Eventually, it takes a little bit of time, but once you develop the adaptive response, then you're going to be able to fight and overcome and destroy the pathogen effectively, eventually, right? But it takes time. So the thing is, you're going to get sick, but you're going to recuperate and then you're going to be fine. Now, once you have this primary adaptive response, the important thing is then your body is like, okay, you got me this time, but you're not going to get me a second time. So you develop what we call a secondary adaptive response, which happens when you get re-exposed to the same pathogen that make you sick the first time. Now, because you already know how to fight that bug, now you're going to eliminate that pathogen very fast in a very efficient way without a lot of inflammation. And all of this because you develop an immunological memory. What I have here is just a bunch of pictures and arrows that illustrate what I just explained a few seconds ago. It's important to look at the numbers because this is the order how I want you to look at those. And the idea is this. You got a disease. I mean, you got an infection. First time exposure. You never encountered those pathogens before. And notice that I'm not being specific. Pathogen is any microorganism that can cause disease. So this could be a bacteria, a virus. This doesn't necessarily has to be in the skin. As you know, it could be in your stomach or respiratory causing pneumonia or just your throat causing sore throat. It doesn't, the same concept applies for everything, for all the diseases. So first you're going to have this group of invaders that will uh, that will get into your body. And what they're going to do is basically use your body, your resources, the sugar, the nutrients to multiply and invade your tissues. As they grow and multiply, they release toxins that destroy and kill your tissue cells, right? So the tissue cells, when they get destroyed and everything, they start releasing chemicals that are inside of the cell, right? We mentioned that those chemicals activate nociceptors, which are those receptors that activate pain, right? But also basically the cells are saying help. We're getting destroyed, right? Because this is the first time you encounter that and you have no idea how to fight or what's going on. The system that will activate will be your innate immune system. Now, I have here just two cells that belong to the innate immune system, but of course, keep in mind that you have more cells, neutrophils, etc. So I have these two because they are one of the most important phagocytes. In other words, cells that deglute uh, bacteria or organisms and then present those to your um, uh, lymphocytes. But anyways, before we get there, look what happens. First, these guys come to fight this new pathogen. So we move to number 3. We can see here that they come and they start fighting. Now, the way how they do that is good because the response is fast. They start destroying some of bacteria, but they're doing that in not in a specific way. What does that mean? Is not specific. That means that they destroy and kill bacteria, but also they destroy and kill your own tissues. The analogy for that is like, think about the immune cells, the innate immune system, like the police or the army. And when they are attacking those invaders, they're using shotguns or they're using grenades. So they just throw that and you have collateral damage. So whenever that happens, yes, it's good because at least you're kind of fighting the infection. But the downside is that all these chemicals that are released and destroy your own tissues just generate more inflammation. What that means is that you're going to have fever, malaise, if it's local, as I said, heat, pain, redness, swelling. All of these are features of inflammation. And that's when you get sick, when you feel sick. So primarily is your own immune system trying to fight. So that is one thing that is happening. And you can see here how these phagocytes are swallowing these microorganisms, right? What they're going to do, so a group of them will stay and fight there, but there are going to be some of them that will take one of those bacteria, and what they're going to do are like messengers. So they will go to present, to tell, or to alert your lymphocytes, the most important ones, that there is an infection happening currently, an active infection. So if you follow me here, I'm looking here, this dendritic cell. The macrophages do the same thing. So they travel until they reach this naive T cell or T lymphocyte. Do you remember what does that mean, naive lymphocyte? We explained this in the last video. So that means that this is a mature T lymphocyte that have never encountered an infection. So it's ready to fight. This is the first time. Okay. So, and remember that these cells, the macrophage and dendritic cells, they're called antigen presenting cells because what they're going to present is an antigen. And what is an antigen? Essentially, is a little piece of the microorganism that is causing the infection. So you can see here in number 4 what happens. We have this MHC, which I'm going to explain it in just a minute, class 2. That little part that we see here is that little arm of this cell. So this is a big magnification, right? And you can see here how this antigen that we can see here is extracted from the pathogen. That is what activates the T cell. And that is the beginning of the primary adaptive response. Now, again, whenever you have primary adaptive response, you're getting sick. But at the same time, now your body is getting ready to fight better, fight back, and get rid of the infection. So once the T lymphocyte gets activated, as we can see here, that's the same picture, right? Two things are going to happen. One is that some of these activation will lead to the formation of memory T cells. Those are the ones that we want because your immune system is kind of saying, okay, you did it one time, you're not going to do it again. So you have the development of memory T lymphocytes or T cells. The second thing is, and of course, depending on the type of infection, I don't want to get specific, but depending on the infection, if it's a viral infection, you might have maybe T cytotoxic lymphocytes more than helpers, or if it's a bacterial, it doesn't matter too much. But what you need to know at this point is that another group, these T helper cells or helper T cells, are extremely important. Okay, those are one of the most important cells because once they activate, they are responsible to activate other cells. Let's look at the first scenario. When this T cell is activated, then we'll release cytokines, and these cytokines are chemicals that will activate the macrophages. Look at how the macrophage of the innate immune system looks like. So it's kind of, it fights, but it's kind of a little bit, a little bit sad, a little bit afraid. And once the macrophage get activated by one of these T lymphocytes, he is like in a ready to destroy. So essentially, a macrophage become bigger, more aggressive, more effective, swallowing and destroying pathogen. So he is ready to go back and fight, in the best possible way. The other important thing is when a T cell is activated, also will activate your B lymphocytes. And those are important ones because once a B lymphocyte activates or gets activated, two things are going to happen. One is that the B lymphocyte will become a plasma cell. And these plasma cells are going to produce antibodies that will target specifically that pathogen. Okay, so these antibodies are very specific for that pathogen. The way how antibodies work, I might describe that later in this class. But for now, you just know that antibodies are things that we need to destroy pathogens. So that is the first thing. And then the other thing, look, you have a memory B cell. So this is going to be important because we have two type of memory cells that we'll see what happens when you have a secondary. Anyways, the point here is that at the end, once the primary adaptive response is complete, that will basically be the most effective way, and the pathogen eventually will get destroyed, and that's how you get rid of the disease or the infection in this case. All right, the secondary adaptive response, if you remember what we just learned, that means that then you get healthy, and then let's say that after a few months, for some reason, you get re-exposed to the same or the previous pathogen, right? So in this case, because you have memory cells, your innate immune system is not going to take, it's not going to waste any time. You already have these memory cells. Once they get exposed to that pathogen, see what's going to happen really quickly. So essentially, what is going to happen is that your immune system will say, "Oh, I remember those guys. We just destroyed them a few months ago." So yes, and we remember what are the weaknesses, right? You just kick it in the leg, punch in the face, and then they're done. So essentially, what happens is your T cells and your B cells, the memories, they will differentiate. In this case, the memory cell will form a T cell that will stimulate the macrophages. Macrophages will become active and then will destroy pathogens. And the same thing with the B memory cells. They will activate B cells, and B cells will become plasma cells, and they know specifically what type of antibodies they need to produce, and that will destroy the pathogen. See what happens is the response in this case is very fast, is very specific, which I didn't add it here, and effective in the destruction or destroying those pathogens. So what that means is that first, you got symptoms. This time, your response is so fast with minimal damage, collateral damage, that you have maybe none or a few symptoms. And then what that means is, congratulations, you're immune to that specific pathogen, to that specific disease. Something that is very important to mention here, which again, I hope that you've been following and understanding this general idea, because we're going to review it in detail next, is that there are two types of immunity. Okay, cellular immunity is or refers to the fact of the T lymphocytes being activated. So that's what we call cellular immunity of the adaptive immune system. And then the humoral immunity refers to the fact of formation or acquisition of antibodies that will fight an infection. So pretty high yield to remember and to know these two things. Humoral immunity has to do with formation of antibodies or creation of antibodies activated by B cells and plasma cells. And cellular immunity is the activation of T cells. Okay, we're going to step back and we're going to start over. Now with that big picture, with that overview that I just gave you, hopefully this next information will make more sense. It will be more easy to understand. First, organization of the adaptive immune system. I told you before, just don't memorize which cells belongs to each one, because the easy way to remember that is just to know that adaptive immune system equals lymphocytes, and then everything else is innate immune system. And what are the lymphocytes that are important? Of course, T lymphocytes, B lymphocytes, and I don't know how many times I said this before now. Remember the difference between a plasma cell and a B lymphocyte. They're pretty much the same thing. It's just that a B lymphocyte gets activated by a T cell and then become a plasma cell, which secretes antibodies. Now, a B lymphocyte also secrete antibodies, but they're not specific, while the plasma cells are specific antibodies that will target specific microorganisms. Right. Remember what I said about how important it is to activate the T lymphocytes. So here you don't need to know this in detail a lot, but just the general idea. In the cell membrane of these T lymphocytes, if we take a little piece of cell membrane and zoom in, we have what we call a receptor. So that means it's like the little arm that will make contact with the antigen that the antigen presenting cell will present, as the name implies. So this receptor just says that these receptors in the T cell, they has two portions. One portion is called constant region, meaning that it never changed. The amino acids that form these proteins here, they're going to be the same. It will not change. And when we have here also a variable region, which is a region that it will change or will be different between receptors according to the antigen. So what that means is it follows the same logic or pattern when we talk about hormones and receptors, or when we talk about antigens, antibodies, that each specific antibody attaches to a specific antigen, and a specific hormone attaches to a specific receptor. Same thing here, and a specific antigen attaches to a specific receptor, and this receptor, depending on which antigen, it will be, it will vary, right? So that's why the region is called variable region. So, but that's all. That's all you need to know. You don't need to know more about this. Again, this is pretty complex when you get in detail and try to get the information very specific, but you don't need to know that. That's what you need to know for your exams. That there's two regions, constant and variable, and then the variable is that one. How these antigen receptor interact, how does this occur? So what we have here, most of the time, an antigen is a protein, and this protein is all this. Okay, so this protein is something that for example, a bacteria might have in the cell membrane. Who knows? Or this protein can be from a virus. It doesn't matter. So what matters is that the whole thing is what we call an antigen. And the other important thing is that when you have an antigen, microscopically, I mean, not even microscopically, but even subatomically, almost at the level of the chemical level, notice that there's a specific parts on those antigens that will attach to the variable region of the receptor of those T cells. And notice that, for example, that's why we have T cell 1 and T cell 2, because again, notice that this is color coded. Notice that this constant variant is still purple, right? And notice that what changed in terms of colors are these ones, right? Those are the variables. That's what that means. So this variable will interact specifically with that part of the antigen, and that specific part of the antigen, which is known as the antigenic determinant, is what we call the epitope. So an epitope is a little piece of an antigen, which is the part that the receptor interact or attaches to. All right, now that you know how this interaction occur, how these receptors and the T cells, how they work, how they interact with the antigen, now we're going to learn how the antigen is produced, how the antigen processing and presentation, right? Presentation to who? To the T cells. So we just learned that your innate immune system, we have those antigen presenting cells that we discussed a few minutes ago, the macrophages and dendritic cells. Those are by definition the classic antigen presenting cells. So you can read these, right? We're gonna go, I'm more visual, so if you like that, I think let's just use the pictures. And everything starts here, right? With a pathogen that your cell will swallow. This pathogen, this bacteria will get inside of a phagosome, which also is just a vesicle, it's a little sac that traps the bacteria. And then you have lysosomes, which are part of the organelles and the cells. And these lysosomes are little vesicles that contain enzymes that destroy things like proteins and fats and bacteria in this case, right? They're like a digestive enzymes. So they get released, they destroy the bacteria, but not the whole bacteria. Importantly here is that, remember, these cells are trying to find an antigen to present to the T cells. So what is going to happen is from that bacteria, from a little fragment. Now, the details about how the cell knows which specific fragment is the one that is going to be presented, I don't even know, and it's not important. That is pretty, detailed. You don't need to know that. Just know that at the end you have a little piece of bacteria, right, with an antigen from digested pathogens, and then you have these receptors. They're called the MHC2. MCH2 stands for major histocompatibility complexes. And there are two types, okay? So the major histocompatibility complex 1 and complex 2 is not super important to get, crazy to know which one is 1 and 2. I'm going to mention them in just in a minute, but just know that this MHC2 is the one that will attach to the antigen. Once you have this complex, then this complex migrates to the cell membrane of the white blood cell, in this case the dendritic cells, and they're ready to be presented. So of course, while that is happening, this dendritic cell is traveling to where, right? If it's a tissue, you have two ways to go, right? So you can go either to the lymphatic system and try to reach the closest lymph node, because that's the place where your T cells are located. Or if this is happening in your blood, the place where you're going to go is to the spleen. And then in the spleen, you have your T and B cells over there. So that's basically how this antigen is formed and completed and getting ready to be presented to your T cells. Okay, so the most important one for you to remember for your exam is the major histocompatibility complex class 2. This is very specific for the immune system, meaning that not other cell in your body have this class 2, but the immune system. So we're going to start with those because we just learned how that is formed, right? So that's what we have here. And then that's the one that will be presented to this naive T cell. And then once the naive T cell interact with that specific antigen, then the T cell becomes active, and then you know what happens after that, right? We discussed that. Now, remember that I told you before, their diseases, they're bacterial and viral. I mean, viral infections always. The way how a virus, just in general, so you know, the way how viral infections invade your body is that they invade your cells. So basically, a virus is not really a living organism that we know. They're just little pieces of DNA proteins that they have mechanisms how to enter the cells and then use the cells, specifically the ribosomes and the nucleus, to make more copies of themselves, more copies of the virus. And then they make more and more copies, and then they leave the cell to invade other cells. But what I'm trying to tell you here is that viruses are intracellular, meaning you will find them inside of the cell, but also in some point you're going to find them outside of the cell when they're going to invade other cells. So you can find it in both. Bacteria, the same thing. There's the vast majority, or the majority of the bacteria, they're extracellular. So that means that they're outside of the cells and they just compete for your resources and kill the tissues by releasing toxins. But there's another group of bacteria that might live inside of the cell, so it might be intracellular. And when you have those types, right, when you have certain bacteria and they are intracellular or viruses, they are going to invade your tissues And the cells that are infected, those are the cells that are going to express the MHC class 1. So you can see here how this is formed. Sometimes these MHC class 1 are particles that has been destroyed and your tissue cells capture them. It doesn't matter the complex. The point here is that at the end, what this infected cell is going to present to your immune system is those antigens, and that is the way how the cell will know, or your immune system will know, that a cell is infected. In addition to that, as we mentioned before, the cell also releases specific chemicals, that interferons, that are released and alert the cells that they have, the immune cells, alert them to tell them that there is a intracellular infection. And in that case, the type of lymphocytes that you're going to need are going to be the cytotoxic lymphocytes, which they work different than the T helpers, and we mentioned that before. So if you don't remember, go back and review. Cytotoxic lymphocytes or T cells are the same thing. Depending on the stimulus, depending on the activation of the naive T cell, the activated T cell will divide or will proliferate in different clones or expansion of clones. So what I want you to look is that these are the T cell clones. So that means is that when a T cell activates, we'll have either 4 types. Cytotoxic T cells, which I just mentioned before, pretty important for viral infections and bacterial infections that invade intracellular tissues. That's so, and remember how they kill, how destroy. I told you, go back and review exactly what kind of chemicals are the ones that they release to generate or to promote, stimulate apoptosis in the tissues. Also, memory cells, pretty important, because remember that is going to be determined for you to not get sick twice with the same disease. The helper cells, which we learn how important they are to activate B cells, and another type of cell called regulatory T cells, which their function is not completely understood, but it's believed that the regulatory, that's what they're called, regulatory, is that whenever you have an activation of your T cells and your immune system is getting crazy, making more copies to fight the infection, the regulatories are the ones that is, they say, okay, don't get too crazy, don't make too much. Kind of regulates in some degree that your immune system don't overreact or overgrowth for something. Okay, but again, that is less important. By far, the most important ones that you need to remember are these 3. But keep in mind, this will be easy if you see the name regulatory. Regulates in some degree the expansion and the aggressivity, how aggressive is going to your immune system will react. We mentioned this before, so I don't think that it's worth it for me to invest a lot of time. These cytotoxics, again, will target bacteria that live inside of the cells. I will give you an example just so you can have an idea of, because again, when you learn this stuff, you need to have in your head, okay, how this is relevant for real life situations, right? Because otherwise, the information in your head will not be relevant. That's why my daughter doesn't want to speak Spanish, because it's just me talking to her in Spanish, and she cannot use it because at school everybody else speaks English, so it's not relevant for her. So her brain denies to learn, right? So it's kind of the same thing with this stuff with anatomy. You have to find a way to tell your brain why this is relevant, so the information will stick in your head. So, for example, viruses, we know all the viruses are intracellular. So, for sure, you need cytotoxic T cells. Another one, cancer cells. Okay, a bunch of cancer cells the way how your immune system destroy those cancer cells and you don't develop cancer is because you have T cytotoxic cells that can react and can detect that a cell is cancerous and will destroy it. Another is bacteria. What type of bacteria? Well, I'll give you just one example, so we don't spend a lot of time here. Is Neisseria gonorrhea, which is a bacteria that causes sexual transmitted disease in males and in females. And males is characterized by all these purulent discharge. If you're interested, you can look at gonorrhea, and you will have all kind of horrible pictures about this bacterial infection. The point here is that bacteria, the way how they grow is by invading and living inside of the cell, and they will multiply inside. And then there's other types. There's parasites that also invade cells. The example of these ones, for example, is malaria. Okay, malaria is, as you probably know, is an infection. It's a parasite infection that is transmitted by mosquitoes, and those mosquitoes will bite you and just get blood from you. And then this parasite lives in the saliva of the mosquitoes. The parasite enters your bloodstream and they invade red blood cells. So the parasite basically uh, uh, lives inside of the red blood cells. So that's an example of an acidic disease in which we need these cytotoxic cells to kill or neutralize them. Memory cells, I don't think there's worth to mention more than what we just covered already. We also, again, we cover this. Don't worry about TH1 and TH2. That is, again, advanced for you. You don't need to know those details. I'm just going to tell you this just so you know. T refers to lymphocyte. It's a T lymphocyte. H, helper. And then 1, because you have two types. The type 1 is the one that activates macrophages, and type 2 is the one that activates B cells. Cool. So let's now talk about the B lymphocytes in more detail. And here you need to know specifically the type of antibodies. And we're going to talk about immunity, and specifically what we call humoral immunity, active, passive, artificial, and natural immunity. So this is a pretty high yield concept that you need to understand more than just memorize, because you have a bunch of questions in your exams that will require more critical thinking, and just put your head and think and comprehend to get to the answers of the questions. Okay, when we have B cells, the most important thing to remember is antibodies, and we've been repeating this so many times that I'm going to be very quick here. And recall what just I mentioned. Maybe this is the second time that I will mention here. Humoral immunity. You have to connect these three things whenever you hear the word humoral immunity. 1, antibodies. 2, B or plasma cells. That's it. If you connect those things together, you're going to get most of the questions right. Cell immunity or cellular immunity refers to activation of T cells. Humoral activation of B cells with the production of the important antibodies. The most important information here is that the most important way, or the primary way how a B cell activates is depending on T cells. So they need T cells in order to become active and differentiate in plasma cells. There are other ways that T cell independent antigen activation, which I don't recommend you to get crazy trying to know a lot about these, because again, this is at least more superficial. Know that the most important part is that T cells are the most important in the way how B cells get or become activated. So this is the most important part, I will say, because now we're going to talk about antibodies in detail. We haven't talked about antibodies in detail, but you will see that it's easy. First, we mentioned about the receptors and the cell membrane of the T cells. Antibodies are very similar in some degree. What we have here is a representation of an antibody. They might be different. Some antibodies are bigger, others are smaller, some more complex. What you need to know is they're proteins, okay? All these antibodies are proteins. And notice this, their antibodies, we have a constant region and they have a variable region. Now you know what the variable region means, and I'm not going to invest more time repeating that. So variable region of antibodies. Why they are important? What is the function of these antibodies? Basically, or in general, two basic functions. One is that they act as a B cell antigen receptors, okay? And two, which is the most important, I will say, is that they can be secreted into the blood, circulate, and bind to pathogens. So that is the good thing, or that is why they're important. Because just imagine that you have a parasite, right? Let's put P for parasite. And you have a B cell or a plasma cell that is producing antibodies. And these antibodies are like things that will not stick to anything because again, you remember antigen, antibody, they have to match each other like two magnets. So when an antibody reaches the antigen that is present in the bacteria, it will attach to the bacteria. In the same way, how when we learn blood, how we learn about agglutination, which remember when there is a incompatibility or when you have antibodies against specific blood types, how they agglutinate red blood cells, in other words, how make them to stick to each other. Well, it's the same thing with bacteria. When you have parasites and you have uh, antibodies that target them, they're going to be trapped or they will not be free basically. They will agglutinate, and then your big macrophages, right, is going to be way easier for a macrophage to deglute all of these ones together. Then imagine if those bacteria are moving away, right? They're like, oh, help, help. One of them might go that way. Others will go that way. Others will go that way. So it will take more time for a macrophage to chase those bacteria one by one. And that's why antibodies are so important for the more efficient way how your innate system will destroy these bacteria. Because again, they stick to those bacteria, they target the bacteria, and that is the way how they work. Now that you know how they work, you need to know the types of antibodies. Basically, we have four types. And for the purpose of your exam, you need to know the most important antibodies, where you can find them, and all of them, they present or they provide some degree of immunity. So, for example, and remember, each antibody is specific for each pathogen. What that means is you can have, for example, let's say that we have three bacteria. So I'll put here bacteria number 1, bacteria number 2, and bacteria number 3. Okay? You have antibodies, IgM, which again, Ig stands for immunoglobulin type M, immunoglobulin type G, blah, blah, blah. What I'm saying is IgM, if you have an IgM that is specific for this bacteria number 1, you can have tons and tons of IgMs. They will not react or will not do anything to these bacterias number 2 and 3 because again, those bacteria might have a different antigen and they will not react. So when we say, for example, I have or somebody has COVID and says that person has immunoglobulin or IgM for COVID is different than have IgM for hepatitis A or IgM for hepatitis B, right? So keep in mind that antibodies are very specific. These types, specifically IgM, are the ones that elevate as a primary response. So what that means is that if you have and then to explain this actually, also let me talk about the IgG at the same time, because the IgG is the um, the main antibody in late responses. So what that means is, in going back with COVID, because you will get familiar with this or you will remember this if you ever heard about it, you will understand now why. When we say IgM is acute and IgG is more late, that means that if somebody tells you that they have antibodies against COVID, IgM, what they're telling you is that they have the disease, or I mean, currently, right? So currently, they have, and their body is fighting against COVID. Now, or somebody said they have IgG antibodies, that means that they already fight COVID, so they're not sick anymore, and those IgG are memory antibodies, um, or that they got vaccinated and they have IgG. So IgG means that you have immunity. So what that means is, again, late protection. All right. What else about the IgG? From all the four types of antibodies, IgG is the only one, because if you look at the picture, see how big and complex is the IgM. So really, it's a lot of antibodies. So it's a very large molecule, while an IgG is just a monomer, so it's a smaller, and therefore this is the one that can cross the placenta, and by crossing the placenta protects the fetus from diseases that can infect the baby. So through these IgG that are produced by the mother, the baby gets protected. And this is going to be important when we learn about immunity, because we have natural immunity, artificial immunity, and then active and passive. We're going to learn that in just in a minute, but I like to tell you that already as an introduction. Which other antibodies are important? IgA. IgA are crucial. They're they're very important because those are the ones that see where we can find these IgAs. So we can find it in secretions like mucus, saliva, tears, basically, and also in the digestive system. So why this is important and relevant? Because the mother produces IgA, and those IgA are present in the breast milk, specifically in that first breast milk called colostrum, that is the first secretion of milk that comes from the mother after birth. So that colostrum, that's why it's so important for the baby's health, right? Because formula do not have antibodies. And this colostrum has these antibodies that the mother has formed, and they serve to protect the infant from diseases. So pretty high yield to know. IgA is present in the mother's breast milk and protects the baby after the baby is born. When the baby is not born yet and is in the uterus, the main antibody that protects the baby is IgG. Okay, so IgG. And remember, you can have thousands of subtypes of IgG that are going to target a specific pathogens, right? So just keep that in mind as I explained before. Lastly, we have the IgE. The importance of the relevance of this is that this IgE is associated with allergies, okay, with allergic disorders, specifically with mast cells. And what we can see here, or the importance or the relevance of this, is that there are people who has severe allergic reactions. So these IgE are the ones that we can find in disorders called anaphylaxis or anaphylaxis, which is an exaggerated response of the immune system that can provoke that the granulation of these mast cells. And if you look at this picture, this is what happens actually. This is a mast cell. The mast cell has antibodies attached to the cell membrane. The constant region is attached to the cell membrane, and the variable region is the one that is exposed, right? These are type of IgE. And then these cells, for example, if these mastocytes are, or mast cells are in the respiratory system, somebody who has allergies to pollen or dust or whatever, people with asthma, for example, right? They will inhale the allergen or the thing that causes allergy. And once that allergen, in this case, will be, it's like an antigen, um, binds to the IgE of the mast cell, that triggers the granulation of the mast cell. And what is this degranulation? What are all these chemicals? Well, are chemicals that generate inflammation, histamine, or other vasoactive things that will basically, to not get too technical, it will just increase inflammation. And just imagine what happens to somebody with asthma when you have inflammation of the airway, you have an excessive amount of mucus, you can have bronchoconstriction, the airway will get partially occluded, and the person will develop symptoms of asthma with cough, shortness of breath, etc. All right, so I mentioned before, we're going to review active versus passive immunity. Remember that when I explained the secondary adaptive response, I'd say, congratulations, you're immune, right? That's what that means. Immunity, or being immune, is that you have memory cells, you have, but in this particular case, that you have memory B cells. In other words, you have antibodies. So that is going to be a very important thing. So look, I'm going to mark here so you can see that all this is about antibodies. And if you have it or if you don't have them, okay? If you have them or not, that's one thing. And the other thing is, if you have them, how do you get them? Do you get the antibodies because your own body produce them, or did your own body synthesize and create them? Or do you have antibodies because somebody, or in some way you receive those that someone else produced, right? It will make sense now when we see it. So very easy, okay? Active versus passive immunity. We're talking about antibodies. So active immunity means that you create or created your own antibodies. That's whenever you see active immunity, that means that you have those antibodies because your immune system um, synthesized them. Okay. Passive, you receive, that's the key word here. Okay. So in passive immunity, you receive antibodies that somebody else create. Okay. So that is passive immunity. Now, you can have passive immunity and active immunity in two ways, natural and artificial. Notice that both have natural and artificial. So to explain the difference here, we need to look at this table. The more important, I mean, you have two options again. You can just memorize this, or you can think about it and not to memorize and just to think and get to the answer of a question by using basic concept and applying common sense. So the only thing that I will say that you need to memorize is that active means that you made use antibodies, and passive you're receiving it. So when we say that you have active natural, natural means, and those are my words, means that you're not um, that you have antibodies or you receive antibodies without a human hand or without a human intervention. So you get them naturally. You will see what that means, human intervention, because that refers artificial when a human hand, is involved. So active natural means, for example, you getting sick. That's an example of this is the adaptive immune response. It's like what we described at the beginning of the class. When you get exposed to a specific infection, you get sick, you suffer, you get fever, malaise, blah, 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 and then your immune system get activated and then eventually start forming an adaptive immune response, which at the end of the process you're going to develop antibodies against that pathogen. If you get infected by the same pathogen again later, now you have immunity, so you're going to be immune. But the reason why you're immune is because now you have antibodies that you created, your own immune system synthesize them, and naturally, because the way how you got it was by getting sick. Let's contrast that with active artificial. Okay, the principle here are vaccines. So what is a vaccine? And I'm not trying to get so technical, but I'm gonna just give you a general idea how vaccines work. Or there are many types, but the point of the vaccine is to create or to mimic an infection. Okay, for example, whooping cough, which is a bacterial infection. All right, um, in this bacteria, there's a vaccine against that pertussis or the whooping cough, which what you have there is that a little piece, the bacteria, which we know that will be an antigen, be inoculated into your blood. So when you get the vaccine, what you're going to have is all these antigens, and your body is going to detect those foreign antigens and will create antibodies with the advantage that you're not getting sick really. You might get a little bit of inflammation, a little bit of reaction, but you definitely will not get sick. Like if you just imagine if you got antibodies by getting sick with pertussis or getting the vaccine and just having a little bit of sore in your arm, and that's it. At the end, with the vaccines, you're going to have formation of antibodies that your own body will create, but the way how you create it or the way how your immune system was stimulated is by antigens that were inoculated by vaccines, and because vaccines are made by humans, that's why those are called artificial. So this is an example of active artificial immunity. To finish this part, let's talk about passive. Passive means we're not going to make any antibodies. We're going to receive antibodies that somebody else create. And we talk about these two examples, placental antibodies. Those are the ones that the mother passes to the baby, and the breastfeeding. Now you say, well, there is human intervention there in the sense that naturally, this artificial refers that you're modifying or you're playing, uh, doing all this stuff in a laboratory, right? These ones are antibodies, are things that are natural. So that's an example of passive uh, natural, right? The antibodies that the mother form and pass that to the baby. So in this case, the baby is the one who has passive natural immunity that received through the placenta or that received through the breastfeeding. Remember, placenta, specifically IgG, and breastfeeding, specifically IgA. Lastly, we have the immune globulin injections. So what is that, right? I'll give you an example, and we can use COVID. Let's say that we have this person here, and we have this, I'm going to use 3 actually, and you're going to tell me. Okay, so we have COVID-19, all right? Let's do the easy one. This guy, he got vaccinated. So therefore, he got what type of immunity? He will get an active artificial immunity because he got the vaccine, his body make the antibodies, he's covered, hopefully, if he got the the vaccine that covers that specific strain of COVID, he will be protected. All right, what about this other one? Let's say that these 2 guys are not vaccinated, and then one of them get infected with COVID, okay? So COVID infection. So the guy will be pretty sad, right? Will be sick, miserable, but then eventually he will recover, he will get rid of the viral infection, and eventually he will form antibodies, specifically IgG, because IgG are the ones that you get once you recover from the disease. Now, initially, in the acute phase, what you develop is IgM against COVID, which eventually get replaced by IgG. So once you recover, then you have IgG. This person has IgG in their blood. All right. Then let's say now this other person gets sick, and unfortunately he has hypertension, he has diabetes, he is old, he has coronary artery disease, had a heart attack 10 years ago. He's a weak patient, so he get very, very, very sick at the point that he's almost dying. So what an immunoglobulin is antibodies. So what is going to happen is, or what happened with COVID, is that persons who got COVID disease, they will get blood. So they will take blood samples from those patients to subtract IgG from those people. And then, of course, all of this is in a laboratory. And then these IgG antibodies will be transferred to the person that is currently sick, with the idea that this person, these antibodies, will help to get rid of the infection and at least faster, right? So that is an example of immunoglobulin injections. That's a passive because this person did not make antibodies, and it's artificial because, again, human intervention took place in that example. As you know, there are diseases that are uh, worse than others. There are viral infections that we have vaccines, and there are other infections, viral infections that we do not have vaccines. We have a flu vaccine that you might get uh, during the year, and then you will still have the flu, and you might say, "Why? Because I just got vaccinated, right?" So basically, this part of the class, just we're going to very briefly just to talk about how these organisms can evade or evasion of the immune system. One way, and one of the most important ones for viruses, is mutation. Okay, so, and that's what I was telling you about the vaccines. Mutation is when there's a change in the molecules that were antigens. When you develop an antibody, remember that they specifically attached to a specific portion of an antigen. Virus, they mutate a lot, meaning that their antigens change a lot. And because antibodies are very specific, if you have a mutation that is big enough, they will completely change the shape and the form and the parts of the antigens then the virus will not be destroyed by the antibody. So that's what that means, mutation. And this is primarily with with viruses, right? That's why for the flu, common cold, that's why there are no vaccines for the common cold, because the cold is too immutase is too unstable. So immutase replicates, and every time that invade a cell and when it comes out is totally different. So it's very hard to make vaccines for those type of viruses. However, there are other viruses that are more stable, and vaccines exist. Think about poliovirus, or the typicals that you get when you're a kid, right? The um, viruses that varicella, for example, or viruses that do not change, even the human papillomavirus, those are ones vaccines that are available, and they provide long-term protection because the viruses do not mutate a lot. So once you develop antibodies, you will become um, immune, right? Good thing about the hepatitis B and hepatitis C, or and hepatitis, I would say A vaccines, that they protect you against those viruses that target the liver. Okay, especially hepatitis B can be bad because it can lead to cirrhosis or cancer in the worst case scenarios. Other ways how bacteria also evade the immune system is by protecting the wall, the cell wall. In microbiology, you're going to learn that bacteria has what we call a capsule, which also is important for that. And you can read other ones. Some bacteria, the way how they evade the immune system initially is by invading the cells, so meaning getting inside of the cells, or tuberculosis, which is very TB. It's a pretty important because TB is a bacteria that your immune system, and that's the picture that I have here, is we have here, and this is a real picture, right, from electron microscope, that this is a macrophage. And hopefully you can see here the nucleus and nucleolus. Well, what you're going to see here is that all these little things, they are inside of this macrophage, are bacteria, tuberculosis, bacteria that cause TB. So Mycobacterium tuberculosis are these bacteria. The way how they handle, or the way how they survive inside of the macrophage, is because these TB bacteria, they prevent that the lysosomes, remember those little sacs that has the enzymes that fuse with the phagosomes to destroy the bacteria. The way how these TB bacteria protects themselves is that they inhibit that fusion. So basically, they live inside of a macrophage. That explains why sometimes people develop, uh, latent or late tuberculosis, or some people they will never get rid of the disease fully, and when they become immunocompromised, the infections can reactivate, and the bacteria lives still there. That's an example of a bacteria that can sometimes evade the immune system by getting inside and preventing um, digestive enzymes from macrophages to be released there. The last one is pathogens can cause or produce immunosuppression. And the classic example I mentioned before many times, HIV, right? Um, this virus, the thing is, this virus target specifically your T cells. And this virus invade and destroys your T cells. And so as the virus replicates and start making more and more and more copies and keeps destroying more and more and more of your T cells, then your T cells account start dropping and becoming less and less and less and less, at the point that you have very few T cells. And then when you have a very low account of T cells, what is this called? AIDS. So the reason why you die is because there are other infections then normally you will take care of them very easily. Now those infections that were like a joke for your immune system to kill, now they cause a lot of problems. Uh, they're called opportunistic infections because now you don't have your most important cell in the immune system, and therefore your whole immunity just goes to the trash. And then people with AIDS, they die because secondary infections, and that's why this HIV virus is so dangerous. So it targets the most important cell. And that's why also there's a lot of conspiracy theories about this virus that was lab made. I don't want to, say that is true or not. I'm just saying that is something that people talks about, how these viruses specifically target the most important cell in your immune system.